Good. Can you guys see the slides? Awesome. Great. So today we're talking about genome evolution near and far. We're going to talk about genome assembly, genome alignment, genome duplication, and then recent human selection, and maybe touch a little bit more on the human migration, population differentiation. And these are some of the awesome folks who've contributed slides. So here we are in the second lecture of Module 5 on comparative genomics and evolution. We have 2 more lectures before the quiz, and then we're talking about current research directions. So the first lecture of comparative genomics, we talked about evolutionary signatures, namely how do we use evolution to understand genomes. And today we're talking about how we can use genomes to understand evolution. And then next week, we're going to be talking about phylogenetics and phylogenomics. So this is really the golden age of comparative genomics. We basically now have 200 plus mammals. We have 10,000 vertebrates. We have an enormous amount of resources for comparative genomics, given the decreasing costs of genome sequencing. So that allows us to now start understanding a lot about genome evolution. So what we're going to be talking about in just a sec, let me turn off my... There you go. We can basically talk about how we can do these analyses. So number one, how do we build assemblies from these genomes? How do we then align entire genomes? We talked about aligning small segments at a time and about blasting and about searching reads, but how do you do whole genome alignment? And then we're going to talk about mechanisms of rapid genome evolution and some evolutionary jumps, and in particular, a very cool story of whole genome duplication. And then we're going to talk about evolution in much shorter timescales, and specifically human selection at multiple timescales. So again, this is when the human genome was first being assembled. This is basically the story. There's 3 billion pieces for those who love puzzles. And this guy's saying, oh, I think I found a corner piece. So how do we assemble genomes? So as I mentioned earlier, sequencing can only sequence a few hundred nucleotides at a time, or nowadays with next generation sequencing, only on the order of 100 nucleotides at a time. So that basically means we can't just simply sequence from one end of the genome to the other. There are some emerging technologies like PacBio and Nanopore that are basically either shifting the nucleotide sequence through a tiny little nanopore and then measuring electric current for pairs of bases, and then from that inferring what was the underlying sequence, or other technologies such as PacBio. But the most affordable technologies basically can only cut the genome into small chunks. And then even from those small chunks, we only sequence 500 or 100 nucleotides at a time from either end of these chunks. And the reason for this is that as the fragments get longer, it's harder and harder to tell the molecular weight of sequences of length 99 versus 100, 120 versus 121, and the signal becomes much less clear as the reads get longer. So one common strategy is to basically sequence the genome with paired ends that basically allows us to infer roughly the known distance between 2 ends of the same fragment. And we can do that at lengths of 10 kb or 2 kb or 40 kb and so on and so forth. 40,000 base pairs, 10,000 base pairs, and so on and so forth. So that allows us to now start overlaying these red sequence fragments and building a scaffold knowing their approximate distances. So who's with me so far on this whole concept of we're first breaking up the genome into many, many pieces, and then we're sequencing both ends of these pieces in order to infer the relative distance between them. So by basically doing fragment size selection at 2 kb or at 10 kb or at 40 kb, we know roughly the sizes of those fragments. And therefore, when we have the 2 ends, we know how they are relative to each other. So we have 9, 0, 0, 0. So now what we'd like to do is assemble these reads. And the first approach and traditional approach is to overlap, lay out, and then build a consensus. So the first step is finding overlapping reads. That's basically, either blasting or aligning or hashing k-mers from one read and another read together, so that you find good pairs of reads and you progressively join more and more pairs, and you eventually build these contigs for contiguous segments. So now these contiguous segments are made out of reads that were directly overlapping. But between them, we know the order and orientation and spacing between them because of these links, because of these paired end sequences. So that allows us to now start building contigs and linking them into super contigs or scaffolds. And then we can infer the contiguous sequence between them, and then insert missing letters as N, which stands for any nucleotide, the letter N. And we can use that to then construct these scaffolds in black with letters in red, and then N's in between, and then letters again, and so on and so forth. So let's see who's with me so far. Lovely. So we have 70, 30, 0, 0, 0. So we can now start asking how do we do all that? So basically, how do we find overlapping reads? Well, we can sort all k-mers in the read, say with k equals 24, and then find pairs of reads that share a k-mer, and then extend those to a full alignment, and then throw away that pair if it's not more than 97% identical. And the reason for this is that there are duplications in the genome, and some of those duplications are much lower identity. So we would like to merge together things that might have either sequencing errors or polymorphisms between different humans, but we don't want to merge together things that are from duplicated regions of the genome. So if they're not very, very similar, we just throw away that pair. We then build these reads into contigs with the concept that we can only extend up until the repeat boundaries. And the idea is the following. I'm building this red contig, and I'm adding more sequence. And then I realize that as I'm adding more and more sequences, I'm entering a region here which is duplicated, which is nearly identical between the blue segment of the DNA and the red segment of the DNA. So basically, two different chromosomes contain, for example, myoglobin gene that is there in almost identical copies. And that is a region that we call over-collapsed. Basically, this is correctly collapsed, this is correctly collapsed, but that's over-collapsed because you're collapsing together regions that are actually separate on the genome. So we basically merge the reads up to potential read boundaries, repeat boundaries, and then we basically maintain those repeat boundaries in a graph that basically tells us here are the unique portions, and then here are, for example, the repetitive portions, and so on and so forth. So basically the nodes of the graphs are reads, and the edges are overlaps between them, where we know the the read identity, and then the shift and the orientation and the score of the match. And then we can remove transitively inferable overlaps from A to B to C, and then resolve these. So the question is, how do you resolve this? If your graph says, oh, here's a unique region A, and then I'm entering potentially an overcollapsed region X or X prime or X double prime, and then I'm exiting that overlap region, and then I'm entering this B unique region, and then I'm entering X double prime again, and then I'm entering C. So A, B, and C are unique regions, but X and X prime are overcollapsed regions. So basically, this is a region that has twice the coverage, and all of these regions were basically built together. So for example, this might have, 10x coverage, This might have 20x coverage. This might have 10x coverage, 20x, 10x. So all I have access to is the fact that A has 10x, 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 and this has 20x. So that basically means that this region is probably twice in the genome, and I have to unfold the graph. So what I basically need to do is traverse the graph in a way that allows me to resolve this ambiguity and eventually take half the reads from here and then lay them out there, and half the reads from here and lay them out there. Who's with me here on resolving these overlaps and unfolding this graph? So we're basically building this graph together, extending these contigs and scaffolds up until repetitive region boundaries, and then unfolding them based on how we can traverse that graph. All right, so 30, 60, 0, 0, 0. Okay, so of course, repeats that are shorter than the read length are perfectly okay, because I basically have, a non-repetitive region in one end and a non-repetitive region in the other end. But repeats that have more base pairs than differences, more base pair differences than sequencing errors, these are also okay. So basically, if the repeats themselves have some differences, and if the sequencing error rate is actually small enough that I can reliably detect that these are differences, then that's still okay. But what we can do is basically make the genome appear less repetitive by either increasing the read length or decreasing the sequencing error rate, basically sequencing at higher quality. And of course, error correction is extremely important because it can detect and discard 90% of single letter sequencing errors, decrease the error rate, and decrease the effective repeat content, because by having, say, 99% accuracy, you can resolve repeats that are only at 97% similarity. But if your accuracy is only 96% and your repeats are 97%, you can't tell them apart. So that decreases the effective repeat content and increases the contig length. And then once you've built all of that and you have the scaffolds, you then want to derive a consensus sequence based on the multiple alignment from the pairwise read alignments. And every consensus base has some weighted voting, which is one way to do it. Another alternative is to take simply the single letter that has the maximum quality, or use some weighted voting based on the quality. So that's the first approach, where we basically first build the scaffolds and build the contigs and then lay them out and then build a consensus. The alternative approach, which we talked about in the transcriptome assembly, is the string graph manipulation. So the consensus layout approach is basically the way that initially the human genome was sequenced and assembled. And then during that time, there was this emergence of new string graphs and de Bruijn graph algorithms for doing this. And then subsequent genomes were aligned mostly through that. So the concept of the string graph is that you can basically reason about tours of how I can traverse these graphs. And I have either one alternative or another alternative, and I can use the long range reads to basically resolve which of these two alternatives is correct or not. So basically, given a shotgun data set of reads, shotgun basically means sequence the entire genome simultaneously rather than go one region of the genome at a time. So shotgun data set of reads, we should be able to build a graph that looks like this and then resolve it into these tours. So the question is, how do we build such a string graph? We can basically use a method very similar to the string layout method, but now we are actually trimming the reads down to individual pieces and building the graph directly from the reads rather than building these contigs and overlaps first. So we can build this graph and then reason about the graph and then collapse long chains of the graph into just one bifurcation event and another bifurcation event, and so on and so forth. So basically these long chains are built out of individual reads rather than contigs now, which allows us to reason about the graph directly from the beginning. So the orientation also matters. Basically, this is a bidirected graph. Basically, forward reads can be distinguished from reverse reads, and the orientation of a piece actually matters greatly. because every single time we sequence, we prime from the 5' end, so we know the orientation of these two pieces relative to each other. So that basically means that we can use the directionality to help resolve the graph, and we can reason about flows into edges and out of edges. We can basically say we want all of the edges coming into any such graph to basically have equal weight as the ones coming out. So basically, if there's a flow of, I don't know, 20 depth here, 20 depth here, and 20 depth here, I better have 60 depth coming out of all this and 40 depth here, and so on and so forth. So I can reason about the flow through each of those nodes. So everybody with me about reasoning about flows? So basically the total sequencing depth coming in and coming out of a particular repetitive region must be the same. So we can actually make inferences about these flows and then use that to resolve these graphs. So 25, 50, 25, 0, 0. So basically we can classify edges based on either a high probability that the edge is unique, or no information, or that the edge is likely overcollapsed. And then based on some uncertainty, for example, here, and some uncertainty there, if I have certainty coming here from another part of the graph that has now been resolved, then I know that this must be exactly 1 and this must be exactly 0. So I can actually reason about these flows. And if I know that this must be more than 1, and then these must be more than 0, and these two are exactly 1, then all I can say is that this is at least 2 rather than at least 1. So I can propagate this and sort of using this constrained propagation, flow through the whole graph and sort of resolve effectively these pieces of the puzzle. So reads from multiple places in the genome are chimeric. So basically this actually does happen. So when you sequence DNA, in some cases, there's a read that is an artifact that was actually during your sequencing reaction connected one segment of the genome and another segment of the genome. So you can actually detect those chimeras using this flow reasoning and actually remove them. And of course, some overlaps are missed because of errors and polymorphisms. So the overall error correction algorithm is seeking to build a local alignment between all read pairs using a very fast algorithm, and then fix parts of reads with indels and mutations that are not supported by any read and are contradicted by at least 2. So you can basically infer chimeras. You can sort of figure out places where it's just a repetitive region and it's resolved, or places where there's an incongruence and you have to just simply trim the read. And of course, some errors are impossible to fix. So you can basically achieve a feasible flow by removing the fewest numbers of reads and then add back edges, and then have a penalty for the back edge, which is equal to the number of reads. And then if you have these edges and these back edges that form a cycle, then this edge can be eliminated. So these are, again, you don't have to worry about every single detail, but just understand that you can build these rules and use these rules to sort of propagate the solution through your graph. So with large genomes, there might not be a unique minimum cost flow. So you can iterate flow sorting, add some penalty to all the edges, solve the flow again, and then repeat until there's no new edges. And then, the edges that are labeled in all solutions are required. Those that are labeled in no solutions are unneeded. And those that are labeled in some solutions are simply unreliable, and you can choose to include them or not. And then this is, for example, the resulting graph from one of these species. And of course, people don't want graphs, they want contigs. So you basically produce a set of unambiguous components as your genome assembly. Okay, so this is basically going beyond this, let's align one gene at a time, to let's assemble a whole genome using all of the intricacies and the complications associated with whole genome assembly. So basically, we saw consensus layout and overlap methods, and also string graph methods. Now let's turn to beyond, now that we have the genome, let's now think, how do we align the genomes to each other? How do we resolve corresponding regions? How do we use gene-based global genome alignment? And then what does that tell us about mechanisms of rapid genome evolution? So multiple alignment is achievable through this dynamic programming method that we saw. And with two species, the pairwise alignment took order n squared. And this was basically the traversal from the bottom, left to the top right of this dynamic programming matrix, where every single time we were inferring the score from the nearby neighbors. And this was order n squared for two sequences. Now, for three sequences, you can think of this as a cube where you're basically starting at the bottom and you're traversing the edges of the cube across all three sequences. So if you move up, it's a gap in this direction. If you move this way, it's a gap in that direction. And if you move this way, it's a gap in that direction. Okay, so you're moving through the cube, inserting gaps in each of the sequences, and then traversing this. And this is basically n cubed for three sequences. Who's with me so far on this sort of multiple sequence alignment, which basically traverses, instead of 2D matrix, a three-dimensional cube going through all of these? And then, with a K species, it would be a K-dimensional cube. Okay, so 80, 20, 0, 0, 0. So the challenge, of course, is that this is extremely slow and the memory requirements are just simply enormous. So what we're going to do instead is build these alignments progressively. Oh gosh, I had the cube as an animation. We're basically going to build these alignments by walking up the tree. So when we have multiple species, and we can infer a tree between them, as we're going to see in the lecture next time. So if I have the tree, instead of aligning n species to n species jointly and simultaneously, what I can do is basically walk up the tree and align this one with that one to obtain an alignment here. Align this one with that one and obtain an alignment here. This one with the alignment here, and that one with the alignment here. And then same thing here, walk up the tree to basically infer through a series of pairwise alignments what the overall alignment should be. Is everybody with me here on this progressive alignment? So rather than building the full n to the k hypercube, we're now going to be building only pairwise alignment walking up the tree. Awesome. So 40, 50, 10, 0, 0, 0. Okay. So now, of course, when you're aligning an entire genome, you don't have the luxury of basically saying, oh, I'm going to start at the bottom left, and then I'm going to move and go all the way to the top right. Basically, there might actually be other solutions that capture more of the alignment. So what we're going to do is basically first find local alignments and then chain these local alignments using a restricted dynamic programming. So the idea is we have a local alignment here. I want to move from here all the way there. So what I'm going to be doing is building a dynamic programming matrix only in between those highly scoring pairs. So we're going to find local alignment. We're going to chain together these local alignments to traverse. And then we're going to do a restricted dynamic programming only with those segments, thereby eliminating the vast majority of our sequence, where we don't actually need to test that space. So who's with me here on this whole concept of a local alignment that is then chained into a global alignment by only constructing the full dynamic programming matrix for a subset? Awesome. So 60, 40, 0, 0, 0. Okay, and then you can build this progressive alignment by moving up the tree in the order of the tree. So that's local alignment. And what you can also do is build a semi-global alignment. So a global alignment requires a single traversal from the bottom all the way to the top. A local alignment doesn't tell you actually how you traverse everything, but a global alignment is sort of a portmanteau between global and local, and allows you to mix and match and choose specific duplication, rearrangement, and other events. So basically here, there's a rearrangement between those two sequences. This one was clearly moved that way. Here there's a duplication. This region is present twice. And here there was an inversion. And we can capture all that and actually include these additional alignments, which would have been missed if I had just constructed something going from the bottom all the way to the top. Could you briefly explain two things? How do you align to an alignment, and how do you generate the hierarchy without an alignment? So we're going to look at both in the phylogenetics lecture, but the key idea is that you can generate the hierarchy by doing the full pairwise alignment of all pairs, and then you can build a tree accordingly. And if you have a whole genome to align, you can always use the same tree, but that you have now constructed once across the entire genome. So we're going to talk about gene trees and species trees in the next couple of lectures. So then, you'll see how we build that. And then the other question was, how do you align to an alignment? So in order to align to an alignment, you basically have, you can extend your dynamic programming score to, instead of only accepting 4 letters A, C, G, T, to basically accept a matrix of values for A, C, G, and T at every location. So I can expand my dynamic programming score to simply, instead of looking up what is the similarity between the C that I have at that position and the G that I have at the other sequence, I could basically say, well, I have 30% of an A and 20% of a C. And I'm aligning that to 6% of a C and 80% of a G. Okay. Does that answer your question? So these are both great questions. So
yeah, thanks. That's perfect.
And you can do the same thing with gaps. You can basically say, well, if I have a gap, I'm always going to penalize it as a gap, and so on and so forth. Okay, so building this global alignment, the goal is to find the least cost transformation of one sequence into another sequence using these additional operations. So we have sequence edits like before, we have insertions and deletions like before, but we now have inversions, we have translocations, we have duplications, and any combination thereof. So basically, here's a duplication, here's an inversion, and here's a translocation, where instead of just moving up, this, it's actually coming after that. So here's, one such result in the local alignment, where you see all of these dots everywhere. And then the global alignment that basically allows you to now go from the beginning all the way to the end with a small number of inversions in that sequence. So we basically now have a mechanism for building these whole genome alignments, which are completely agnostic as to what are the genes that are sitting in these regions. But we can also build gene-based global alignments between pairs of genomes. So this is again seeking to understand how we can use genomics to understand evolution. So again, the goal is to find corresponding regions so that we can align these genomes and infer what are the conserved elements. So to align these genomes, we want to anchor genomic segments by the genes that they contain, and then resolve the correspondence of genes for every pair of species, and then construct the nucleotide level alignment after you have the correspondence. And the information that's available is the amino acid similarity of every gene pair and the locations of genes in every genome. And what makes it hard is that not all regions have a one-to-one correspondence. There's gene divergence, duplication, and loss. There's genome rearrangements. So we're going to have to find ways of coping with that. So the algorithm that we're going to be describing is the BUS algorithm, or the best unambiguous subgroups, to resolve the correspondence of genes and regions using the complete bipartite graph connectivity of which genes are similar to what other genes. So that integrates the protein similarity, but also the gene order information, And these can correctly resolve the gene correspondence with more than 90% of the genes having one-to-one correspondence, but also identifying regions and protein families of rapid change. So these computational tools give us the ability to automatically align complete genomes. So the idea is that if we have one species here, another species here, and we have bidirectional best hits. So the best hit of this one is that one, and the best hit of that one is that one. That is the easy solution. But you can also have one gene here duplicating into two genes there, or one gene here duplicating into two genes there, or even sometimes one gene here being split into two halves, where perhaps there was a Contig break, and that's why we shouldn't be thinking of this as a duplication event. So we're going to use a weighted bipartite graph, which represents the gene correspondence, where the nodes are genes with coordinates, and the edges are the sequence similarity with weights. And we're going to have two types of evolutionary relationships. Orthologs will be one-to-one matches, and then paralogs will be one-to-many or many-to-many. And the method is going to seek to eliminate spurious edges, i.e. simplify the graph, by selecting edges based on the available information and finding blocks of conserved gene order, and also using the protein sequence similarity. So the idea is the following. We're going to basically use this full bipartite graph, which is weighted. So we actually have the scores. We can find locally maximal groups that have all of the best matches contained within them, and use that to effectively resolve that ambiguity. So we're going to extend the concept of best bidirectional hits to capture these one-to-many and many-to-many relationships. And we're going to use an iterative algorithm that basically builds that full bipartite graph, and then uses the directed graph to find maximal out edges given a relative threshold, and then separate connected components of this graph, and then further subdivide after that. So, and then the idea at every iteration is that we increase the threshold of relative similarity between the top hits and the lower hits to basically find these best unambiguous subgroups. So we're basically also going to be preferentially selecting edges in synteny blocks. So the initial set of hits across 6,000 genes and 6,000 genes for two pairs of yeast species is looking like this, where there's a lot of additional extraneous similarity. But after a few iterations, what you can basically see is that the one-to-one hits are mostly falling along the diagonal, which basically means that there's a lot of collinearity. And then the additional hits are appearing as streaks of protein families that are expanded. So you can see here that all of these additional hits are basically amplifications of this, which are found throughout the genome. And you also see that there's a lot of additional duplications that are happening at the boundaries of chromosomal segments. So basically these are chromosomes 1, 2, 3, 4, 5, all the way to 16. And you can see that at those chromosome boundaries, there are in fact a lot of additional duplication events. So what you end up with using this algorithm is very clean picture of what are the genes that correspond in every one location. And in some cases, you can see this breaking up of this gene into two smaller segments. You can see that some segments are lost, others are duplicated, and so on and so forth. So that's for most of the genome. But we can actually now focus on the differences and actually recognize mechanisms of rapid genome evolution. So this is the picture that you see for most of the genome. But as you start approaching chromosome ends, which I mentioned here, have a lot of additional duplications and losses, what you notice is that this alignment basically very rapidly breaks down, and you see these one-to-many and many-to-many matches with multiple genes that are actually members of gene families. And what's really happening is that the chromosome ends are domains where there are a lot of protein family expansions. So in the yeast genome, we found that there's a lot of environment adaptation genes, such as cell surface adhesion and sensing of the environment and so on and so forth, which are actually amplified in gene families and rapidly evolving. in these regions that are themselves fragile. So it's kind of advantageous for the species to lay its rapidly evolving families in regions of the genome that are already rapidly evolving through massive rearrangements, breaking, etc., because these are at the end of chromosomes. So there appears to be some kind of evolutionary mechanism that is allowing these to evolve rapidly. And these are called telomeres. Centromeres are the centers of chromosomes. Telomeres are at the end of chromosomes. So you can see basically these telomeric expansions of protein families. You see these chromosomal exchanges where you basically have these genes matching completely, and then boom, at some point you basically switch from one chromosome to a different chromosome. And then there's a translocation event that's happening here. There's an insertion that you can see readily here. Here's an inversion that, again, you can see is actually flanked by tRNA genes. These insertions are usually flanked by these transposable elements, Ty, and then same thing for these rearrangements. And then another very cool event that was discovered is this intine insertion. This is a protein that is spliced post-translationally. An intron gets spliced before translation. An intine is basically a type of virus that will insert itself in the middle of a protein sequence, and then as that protein gets translated, it will excite itself, guide the insertion of its own RNA into a new location, and then reinsert itself and replicate. So this is a very simple form of a virus. We also found that there's, again, very few changes in gene dosage or very few cases of family expansions, only 3% involving 200 genes. And we also found that there are particular mechanisms that actually mediate these rearrangements. Every time we saw a translocation, it was through repetitive element. So these repetitive elements can be thought of as an evolutionary feature that is helping these rearrangements happen. Similarly, we found that every inversion was flanked by tRNA genes. Again, you can think of them as evolutionary features where these are the building blocks of evolutionary events, of translocations and inversions. So let's see, who's with me so far on these mechanisms of change, which are rapid. Awesome. So 20, 40, 30, 0, 0. So beyond these traditional models of change, which are happening one amino acid at a time, you can basically see that there are locations where there's an entire domain being created. There's like a lot of additional amino acids that are inserted. You also see some examples of compensatory frame shifts, where you basically have a very, very rapidly evolving region at the protein level, which is simply stemming from an addition of one nucleotide here and a deletion of one nucleotide there, effectively changing the frame of translation of this intermediate signal. You also see that in many cases there were stop codons that were being read through, especially in periods of stress. And again, this intine gain, which was gained in Saccharomyces cerevisiae. And these are evolutionary shortcuts, which become apparent when you look at closely related species. So this was basically looking at cataloging every single evolutionary event between 4 closely related species, roughly at the distances of human, mouse, rat, and dog. We can now look at much, much larger evolutionary distances, and specifically across these evolutionary jumps, these very rapid periods of change. And in particular, looking across a whole genome duplication event. So this is work that I did as a PhD student. So basically the first 4 species aligned very, very nicely. And then we had a 4th species. So we basically had Baker's yeast, which was the reference. And we had sequenced these 4 additional species, Paradoxus, Micatai, Baianus, and Walti. And what's really funny is that we had actually requested a different species and they mislabeled it and mistakenly sent us this species, which was much further than we had requested. But we got lucky because it turns out that that species was separated by a massive whole genome duplication event here. So we were for the first time able to see across a whole genome duplication event. So we can now ask a different set of questions by having these distant species. And remember this very nice picture that I showed you before, where the gene correspondence was largely collinear, where it was a nice straight line. Well, I was very proud of this algorithm that I had written. And then what I saw is that this algorithm had completely messed up this pair of genomes. Basically, the Saccharomyces cerevisiae, baker's yeast genome, appeared to be very messy when it was being mapped on this new species, this Clevermyces walti genome. And I promised my collaborators that I would sort of figure it out before going on holiday, before the end of the year. So it was 11 p.m. I was sitting in the lab and I was staring at this alignment and I was like, this makes no sense. What could have possibly happened? So I was zooming in and zooming out in various ways. And in one way was actually zooming in only in one direction. And what I saw was the following. Notice this region here. Every dot is a gene, and the x-axis is the position in one species, and the y-axis is the position in baker's yeast. What you see is that this region here of this species maps twice onto the genome of Saccharomyces cerevisiae. And if you look at how the mapping actually happens, this region is actually not mapping every gene into copies. In fact, there's very few genes in 2 copies. There's the 3rd one here and the 13th one here. All of the other genes are in exactly 1 copy. So what's going on here? So, and this is true not just of this one region, but in every single one of these regions. There appears to be a 1-to-2 mapping where not every gene, all the genes are mapping, or at least 90% of the genes are mapping uniquely. But the genes are actually interleaved with each other. The genes are actually, you know, one is mapping here, the other one's mapping there. The next one is mapping here, the next one is mapping there, and so on and so forth. So that's very strange. So what could be happening? So why would this region here of this new species map both to chromosome 7 and to chromosome 10? But only half the genes in each place, and the genes being interdigitated. The way to understand what was happening, and that was the aha moment that I had at like 11 p.m. at night, and I started emailing everyone, is that this gene interleaving is actually evidence of complete duplication of this region. Namely, this region here was present in 2 copies completely, but this copy lost this gene, and that copy lost that gene. And here both genes kept that, both copies kept that gene. And here this gene was lost in this copy and that gene was lost in that copy. So let's see, who's with me so far? Awesome. So we're at 40, 40, 10, 0, 0. So hopefully the picture will become even cleaner as we go from this visualization where the duplicated, the pre-duplication genome is in the bottom and the 2 copies are at the top, versus the pre-duplication version being in the middle and then the 2 copies being at the top and at the bottom. So what's actually happening is that this region was fully duplicated, and then because the species only needs 1 copy of this gene, it was free to lose that one. And because it only needs 1 copy of this gene, it was free to lose that one. And because it needs only 1 copy of this gene, it was free to lose that one, since it already had that one. So for this gene, it already had the red, so it could lose the blue, and so on and so forth. And if you look at the gene numbers, they're consecutive. So this is yeast, chromosome J, numbered from A through 16. Left arm, and then 024 counting from the center. C is on the Crick strand. So 24, 25, 26, 27, 28, 29, 30, 31, 32, 33. So the genes are in the same, they're actually compact. And these genes are exactly all of them are there, 37, 38, 39, 40, 41. And there's 2 genes that are in 2 copies, and these are in the middle. So this gene interleaving is actually evidence of complete duplication with very few genes remaining in 2 copies. And this interleaving, painting the genome of Pleuramyces walti with 2 colors at every location based on the 2 chromosomes of Saccharomyces cerevisiae that map there, allowed us to now map the entirety of the genome in these overlapping segments. And that's basically when we could say with confidence that there is absolutely no doubt the entire genome of that ancestral species was duplicated in a whole genome duplication event, giving rise to the modern yeast species. And it's a little ironic that we basically have 8 chromosomes in the ancestor and 16 chromosomes here. And in fact, if you look at the centromeres, these are the centers of the chromosomes, the centromeres are again mapping 1-to-1. And the reason why I like this picture is that it shows that there's not a single gene that's actually maintained in 2 copies here. And in fact, there's 2 genes that are lost. And this is showing you the actual genomic coordinates. So you can see here that the intervening regions were just simply condensed and lost. And there's some ancestral genes that were also lost. And the other thing you're seeing is how the centromeres fit exactly, but also that the interleaving is what gives us the evidence of complete duplication. And if we didn't have the gene order of this ancestor, we would never be able to tell that there was a whole genome duplication event separating these regions from each other. All right, so let's see, who's with me so far on the whole genome duplication event? Very cool. So 40, 30, 20, 0, 0. So we can basically recognize the sister regions solely based on the gene order. And now we can paint the entire genome of Saccharomyces cerevisiae folded onto itself. So this is S. cerevisiae versus S. cerevisiae. So Baker's yeast versus itself, where for every chromosome, you can see which other chromosome matches that region. So there's 145 blocks, and these cover 88% of the genome. This is a whole genome duplication event. This is not just a series of segmental duplications, because you would not expect to cover the entirety of the genome and to not, without having a large number of overlapping 3-way overlaps. So basically our model is the following, that there was in fact a common ancestor that had 1 copy of every gene. And what we have now is that common ancestor of K. walti. And then that common ancestor underwent a whole genome duplication, followed by a lot of loss, followed by condensation of the lost regions, leading to the current interleaving of this. So now that you see this picture, let's see who's with me on the clarity of that whole genome duplication event. So interestingly, one of my friends basically sent me a copy of their textbook, and apparently this figure has now made it into their biology textbook, which is kind of cool. Awesome. So 60, 40, 0, 0, 0. All right. So notice that these 3 here and these 13 here, which happens to be my birthday, March 13, are the only 2 genes that are kept in 2 copies, that the vast majority of the genes are in fact single copy in this region. But using this evidence, Ken Wolfe had actually postulated that because of all of these segments that actually had 2 genes that were duplicated, 2 or more genes that were in the same order and the same orientation, despite having completely unrelated intermediate genes, he had actually postulated that there was, in fact, a whole genome duplication back in 1997. So his genomic evidence was the conserved order of paralogous genes. They were in the same transcriptional orientation. However, they were interspersed with single copy genes. So the interpretation that Ken Wolfe made was that, in fact, there was a whole genome duplication followed by gene loss. But Bernard Dujon and many others had basically published a series of papers saying, there was no whole genome duplication, at least some chromosome duplication occur independently, that there's a dynamic equilibrium of duplication and loss, that, there's perhaps continuous block duplications and deletions, and so on and so forth. So basically there's a large debate about this. And eventually, with our paper, this was completely resolved because we actually had the non-duplicated ancestor. So basically the evidence was insufficient. Only 50% of the genome was in duplicate regions. Only 8% of the genes were present in 2 copies. And there was also extensive redundancy outside the duplication regions. But again, Dujon and others raised evidence against whole-genome duplication, suggesting that the divergence-based dating suggested multiple times that could simply be because of the rates of divergence, and also that other species had similar level of redundancy. Only 8% of the genome was in 2 copies. It's very difficult to sort of have the world believe you when you say that there was a whole-genome duplication event. If in fact only 8% of the genes are in 2 copies. And that's why they had proposed an alternative evolutionary scenario of independent segmental duplications, which was also consistent with the evidence. So after a paper on the yeast whole-genome duplication, we had a beautiful collaboration with a French group that basically was led by Olivier Chaillon and others, showing that in fact there was a vertebrate duplication in fish. That in fish, the gene order was not conserved, only the chromosomes, but in mammals, the gene order was conserved, but not the chromosomes. And you can see here that, in fact, this was the whole genome duplication that we found in all of these teleost fish, including fugu. But after these 2 papers, there was another paper suggesting, in fact, 2 rounds of whole genome duplication in the base of the vertebrate lineage, based on clusters of related genes using Sionna as an outgroup, counting duplications by reconciliation, and then finding regions of duplicate overlap with 4-way synteny. So now the current model is that there were in fact 2 rounds of whole genome duplication giving rise to humans. Basically that the region where we have 4 Hox clusters, these are the developmental patterning genes that set up the body plan of the human, and of course all the other vertebrates, are in fact resulting from 2 rounds of whole genome duplication of an ancestral single homeobox cluster that is found in Drosophila and in other species. And that's the 2R hypothesis of 2 rounds of whole genome duplication, which again is somewhat controversial still because the distances are extremely large, but it, is based on the same type of signature. All right. So now having seen these, okay, hold on one second. There's a chat question. Why would tRNAs enhance inversions? So for the tRNA inversion relationship, the model might actually be that, again, this is pure speculation, that basically the tRNA itself encodes this clover leaf structure, which has 3 regions of self-complementarity. So I don't know if you recall from the RNA lecture, but there are, these self-complementary regions. That basically means that when you lay this out completely flat on the genome, you have this self-complementarity here, the self-complementarity here, and the self-complementarity here, which then folds into, A, B, C, D, E, F, G, H, A with H, and then B with C, D with E, and I'm missing one of them, okay? So that basically means that as I have my linear genome here, this region and that region might actually be able to, in the DNA double helix, sort of create a pairing with itself, where basically, you can actually partly open up the DNA and then create a double helix here, a double helix here, a double helix here, a double helix there. So that's one possibility, where basically the DNA itself could be breathing and creating this alternate structure during transcription and so on and so forth. Another possibility is that because these regions are perhaps fragile through extensive transcription, they end up breaking every now and then and just being religated. And then the tRNAs themselves are in fact self-complementary, similar, and they were all in these opposite transcriptional orientations, suggesting that perhaps irrespective of this cloverleaf structure, there might simply be a region here which is homologous to another region here, causing this inversion between them. Okay, does that answer your question? These are all great questions, so you guys should keep them coming. All right, so we talked about this dramatic event of a whole genome duplication and this evolutionary signature of whole genome duplication. We can now talk briefly about the post-duplication emergence of new functions and then turn to recent evolution. So again, as I mentioned, the entire genome of yeast was duplicated. So we went from 5,000 genes to 10,000 genes, but very rapidly, 90% of the duplicated genes were lost, leading to a gain of only 500 genes. And that's the roughly 8% of the genome that is currently in 2 copies. So the question is, what happened during this post-duplication evolutionary event? In particular, we can focus on these 500 genes that were gained in 2 copies and ask what happens after duplication. So 457 genes were kept in 2 copies, and that was probably the result of selection. These are involved in sugar metabolism and fermentation. And if you go in the fields of Tuscany, Saccharomyces cerevisiae is one of the best yeasts out there, one of the best fermenters. And the, its ability to metabolize sugar and to ferment to kill off bacteria who are competing for the same sugar might actually have given it this evolutionary advantage. So the question now is there any evidence of accelerated protein divergence? And this is actually exciting because for the first time, we can actually now root the tree. We don't just have paralogous copies, we actually have the ancestor, and we can tell how much time has elapsed on one branch versus the other branch. So we can actually ask, what are the possible scenarios for rapid gene evolution after whole genome duplication? One possibility is that one of the copies would evolve faster. And this is the model of neo-functionalization that Ono had suggested. And another possibility is that both copies evolve faster, which Lynch and others suggested thereafter. So the question is, how often do you see each of those? And what we see is that in 20% of cases, there is in fact acceleration, but in 95% of those, only one of the 2 copies evolves faster, suggesting that perhaps this new copy, basically one of the 2 copies, takes a new function. So that's a neo-functionalization, as opposed to both of them taking on sub-functions of the original function of sub-functionalization. And there's some striking examples. For example, this origin of replication protein, which actually sets up replication, is highly homologous to this silencing protein, SIR3. And this origin of replication complex, ORC1, is appearing to be the ancestral form. It evolves at the same rate as the non-duplicated genome, whereas the other one shows a 4-fold acceleration, suggesting that this silencing function was in fact co-opted from a duplication of this origin of replication function. Similarly, for translation initiation, we basically have this HBS1 gene, which after duplication underwent 3-fold acceleration and now gave a new gene associated with antiviral defense. So this asymmetric divergence allows us to now recognize what are the ancestral versus the derived functions for those genes. So who's with me so far? Awesome. So 30, 60, 10, 0, 0. So what we're finding now with these ancestral versus derived function, the slower copy versus the faster copy, is that if you delete these genes, and yeast has been tested with many, many experiments, including deletion of every single gene to see if they're lethal. And what we find is that the ancestral function genes are in fact lethal 20% of the time, whereas the derived function genes are never lethal, suggesting that these ancestral function is essential, but the derived function might be more auxiliary. So that also suggests that we gain new functions and we actually lose the ancestral function. Similarly, if you look at the expression of ancestral function genes, they're much more abundantly expressed, whereas the derived function genes, the faster evolving copy, is much more specific in its expression and very often associated with stress or starvation. And then if you look at the localization, the ancestral function shows general localization, but the derived function is actually usually specific localization in the mitochondria or in the spores, and so on and so forth. So again, this story allowed us to both resolve the ancestry of whole genome duplication and massive gene loss in the yeast Saccharomyces cerevisiae, but also understand the emergence of new functions after duplication, and specifically the asymmetric acceleration, distinguishing ancestral and derived functions, and potentially creating a repository for buffering mutations or simply gene dosage in those other 80% of the genes that did not show acceleration. All right, so we talked about building genome assemblies, constructing whole genome alignments, both by resolving region correspondency irrespective of genes, but also gene-based global genome alignment, and how we can use that in short evolutionary distances to understand mechanisms of rapid genome evolution, and also in large evolutionary distances to study these evolutionary jumps, such as whole genome duplication, and understand both the signature of the event itself and the post-duplication emergence of new functions. Let's now look at the near and far, the near part of evolution. So this was the far part and this was the near part. So now we're going to look within the human genome, so within the human population, for measuring selection at multiple timescales within the human. So if you look at molecular evolution, you can basically ask within the human population now, what is the frequency of the A allele and of the G allele in the population? And you can also ask, how frequently do I see the AA genotype, the GG genotype, or the heterozygous AG and GA genotype? So what would I expect given that 40% of the chromosomes, if I take all of the chromosomes at random, are G and 60% are A? What I would expect is that I would see the GG genotype, the product of those two, which is 16% of the time, the AA genotype, the product of those two, and then the AG or GA genotype, 2 times the product of those two, okay? And the fundamental question is, how does allele frequency change over time? And again, under strong assumptions, for example, no mutation, no migration, no selection, large population size, and random mating, you would expect exactly this picture. You would expect that the allele and genome frequencies, genotype frequencies, remain in what we call Hardy-Weinberg equilibrium, which basically means that if you multiply these frequencies, you get effectively these products. So who's with me so far on Hardy-Weinberg equilibrium, and specifically how we can predict the frequency of each genotype just by multiplying the allele frequencies of each of the alleles? So when these match, we basically say that we are in equilibrium. And that means that there's no mutation, no migration, no selection, and random mating. So 40, 50, 10, 0, 0. And again, under strong assumptions, no selection, and a constant population size, random mating, and no overlapping generation, the change in the allele frequency of a new mutation over time is described by this Wright-Fisher process, which basically says over generations, as this new allele appears, will it rise in frequency or will it drop in frequency? And what you can see is that, drift alone can lead that new allele to go back down to 0 frequency, and that's a sync state. So that allele has been lost again completely, so it's not there anymore. Or it could actually go to fixation, and now the ancestral allele is lost completely, and again, it's nowhere to be found, so it's not going to come back. So both of those are sync states. So without selection, the main force on allele frequency is genetic drift. So this is just random chance. And natural selection can change the course of this process either towards 0 or towards 1. So we can use now selection to ask, has there been selection for specific genes in specific populations? and we can measure the presence of selection at different time scales. So the fundamental question is, how have the allele frequencies changed over time? And different methods basically give us insight into the change over different time scales. So in very long time scales, for example, since divergence from chimp, we can basically look at the proportion of changes that are changing the amino acid. How many nucleotide substitutions are in fact amino acid changing? Or we can basically ask, what is the overall level of heterozygosity? Hetero means different. Zygote is like the balance, zygi in Greek. So heterozygote basically means that the two alleles have different identities. For example, an A and a G, as opposed to a homozygous, which is AA, or a homozygous, which is GG. A heterozygous has AG. So the question is, what is the heterozygosity of rare alleles that functions over this time scale? Or whether there are high frequency derived alleles at shorter time scales, or whether there in fact are population differences between, say, Africa, Asia, and Europe, or the length of haplotypes within each of these populations. So let's go through on how do we measure selection at these different time scales. So, quick question: who's with me so far? Do you guys, sort of get the what are we asking? So basically, the question is there selection, or is it just random neutral drift at different time scales for specific alleles? All right, so we have 30, 60, 0, 0, 0. All right, so how do we test for positive selection? Well, what we can do is ask how many changes disrupt an amino acid. So basically, that's a nucleotide change that happens in the codon positions 1 or 2, or in a twofold degenerate position that actually switches between different amino acids based on that genetic table. So we can ask how many amino acid changing mutations are there versus how many amino acid preserving mutations are there. And why do we care about both? Because the synonymous substitution rate tells you about the mutation rate at that location, whereas the non-synonymous substitution rate tells you about amino acid changes. And if there are more non-synonymous than what you would expect, then you can assume that there's probably some positive selection acting in that region. That basically, even though I would have expected, I don't know, 2/3 to be benign and 1/3 to be amino acid changing, I find that perhaps 2/3 are amino acid changing. That suggests positive selection. Is everybody with me on that concept? That basically, if I have the number of amino acid changes be higher than what I would expect based on the mutation rate, which I can estimate from the synonymous, then that is evidence of selection. So uh-oh. Okay. I see, perfect. Oh boy. Can you guys vote again? I'm sorry. There's some connection problems. I hope I don't lose my connection. Okay, awesome. Okay, so we have 70, 70, 20, 0, 0. That's strange, but anyway, it is what it is. Oh, it's because it was a multiple choice. Anyway, so mutations that were beneficial to Homo sapiens after divergence from chimp basically will not be conserved within other primates. And what the McDonald-Kreitman test is identify sites which are under selection within a species. And then the idea is to compare the amount of variation within the species, i.e. polymorphisms, with the amount between species, i.e. substitutions. So you can basically look at fixed positions versus polymorphic positions and ask how many of those are in fact changing synonymous positions versus non-synonymous. How many are amino acid changing versus not? And if the number of amino acid changing positions, normalized for the substitution rate is in fact smaller than what you find at polymorphic positions. And that implies purifying selection. That basically means that I'm changing less than what I would have expected based on the polymorphic mutations. Whereas if the number of fixed positions is more likely to be amino acid changing, that implies positive selection. So fixed are the ones that basically rose to fixation, whereas the polymorphic are the ones that are still somewhere here in the middle. So then the question is, yes, there are fixed sites and polymorphic sites, but if the fixed sites are more likely to disrupt amino acids, then that suggests positive selection. Okay, let's see if you guys are with me here. Great. Awesome. So 30, 60, 10, 0, 0. So again, TS is the number of synonymous substitutions per gene. DN is the number of non-synonymous substitutions. And then PS is the number of synonymous substitutions in polymorphic sites. So that's basically measuring the proportion of functional changes. That's basically DNDS. This is the number of synonymous versus non-synonymous substitutions, and also heterozygosity based on this. So let's look now for additional tests of high-frequency derived alleles. So basically, this is if we have human here with a lot of humans with either homozygous or heterozygous, we can ask, what is the chimp allele at that position? And if the chimp allele has one of the two from human, we can infer that the derived allele is T and the ancestral allele is A. So then the question is, if the derived allele is at 20% frequency in the population, that suggests that perhaps something is causing it to be high. So measuring the frequency of these derived alleles is another sign of selection. If the derived allele is, drifting along somewhere here, that's one thing. But if it sort of has pushed to fixation, even though it's a derived allele, that suggests that perhaps this is an advantageous allele for being a human. Okay, everybody with me here on this sort of ancestral allele versus derived allele and the frequency of the derived alleles, namely the alleles that are different from the ancestral sequence that the chimp relative still has. Of course, in some locations, chimp itself might actually have a polymorphism, or chimp might be missing that sequence altogether, and therefore we cannot evaluate it everywhere. All right, 75, 25, 0, 0, 0. All right, now we can also ask, are there differences between the alleles that the African continent has versus the Asian continent versus the European continent? Namely, are there population differences? And what we're going to be computing here is FST, which is the fixation index. So if you have basically one subpopulation where A is more frequent versus another subpopulation where T is more frequent, you might infer that maybe there was an adaptive, maybe there was an adaptation, that basically maybe this population has an A because, I don't know, they're exposed to too cold of a climate or too high of an altitude and so on and so forth. So we can measure the differences in allele frequency in populations by the fixation index statistic FST. And this fixation statistic estimates the reduction in heterozygosity expected when two different populations are erroneously grouped. So the question is, are these sampled from the same population? So how do you measure heterozygosity? It's basically 2PQ, which is what we saw earlier when you have basically these, the two alleles being at these frequencies. So then when you have this heterozygosity for any subpopulation, you can ask, FST is effectively the heterozygosity across the combined population if I have the heterozygosity measure in the full circle versus the heterozygosity in each of the subpopulations divided by the total heterozygosity. So basically FST is 1 if it's highly differentiated between the different populations. So if population 1, for example, has more A's and population 2 has more T's, then you can basically calculate this heterozygosity in population 1 based on 2PQ, heterozygosity in population 2, 2PQ, heterozygosity in population 3, and then the total heterozygosity is 032, and then the subpopulation heterozygosity is this, and then removing those, you basically have that. Okay, so that's basically looking at population differences between the different continents. So who's with me here on measuring FST, this heterozygosity in the combined population versus heterozygosity in the separated populations? Awesome. So 40, 50, 10, 0, 0. Okay, so the last test is basically measuring the length of haplotypes, and that's a test that's applicable in much shorter timescales. So why is that? So haplotypes are basically these long stretches of the genome that are inherited as a block because of these meiosis events creating these recombinations and these tragic love story of PRDM9 that basically goes and guides the cutting of the chromosomes during this, metaphase, where the chromosomes are lined up before anaphase, where they're separated. You basically have this PRDM9 protein that creates these double-stranded breaks, causing strand invasion, lining up the chromosomes, and then eventually crossover or in some cases gene conversion, and eventually segregation of the chromosomes. So these crossovers happen over time, and eventually, a long segment will be broken up by these PRDM9 recombination events. So what you see over time, as a recent haplotype becomes older and older and older, what you see is that the LD actually decreases. So you basically have a drop in heterozygosity over evolutionary time. And of course, if the so young haplotypes are long, where there's no recombination yet, and then old haplotypes are short. This is a typo, because recombination events break them up. And of course, the rate of breaking up depends on the local recombination rate R of the region in a given population. Everybody with me as to why haplotypes will get shorter over time as more events break them up, with PRDM9 basically making these double-stranded breaks, creating, as the PRDM9 motif appears in those locations, this will be a new hotspot of PRDM9 cutting, and that will basically create these, cause these haplotypes to be broken. So we have 60, 40, 0, 0, 0. So now here's where it gets really cool. If you look at long haplotypes, that basically tells you that this haplotype is young. And then short haplotypes, this tells you that the haplotype is old, okay? So haplotypes move from young to old. Just by staring at the haplotype length of a segment of the genome, I can tell how old it is. If it's very long, that means there hasn't been enough recombination, so it's still young. If it's very short, that means there's been plenty of recombination and it's very short. And now you can relate the age of the haplotype from its length with the frequency of the haplotype in the population. And you can look for low frequency haplotypes, suggesting that there's no selection, or common high frequency haplotypes, suggesting that there's selection. And with no selection, the frequency will increase by drift, or it might stay low by drift as recombination events accumulate. So basically, I might sort of have this haplotype increasing frequency gradually, or just decreasing frequency again. But if a haplotype is both long, which suggests that it's young, and common, which suggests that it's high frequency, that is a sign of positive selection, that the haplotype frequency was increased rapidly, but the recombination events don't have time to accumulate. So young haplotypes start at low frequency, then increase or decrease by drift primarily. And a long haplotype, i.e. a young haplotype at high frequency, is evidence of selection, that there's no recombination yet. And the reason is that some allele, which is carried by that haplotype, is advantageous. And then its frequency of that allele increases very rapidly, bringing along with it neighboring variants on the same haplotype, as recombination does not have sufficient time to break them down. So a signature of recent positive selection is common variants that are high frequency on long haplotypes that are young. So that's a rapid rise in frequency due to selection with no time to accumulate mutations. So let's see who's with me so far. Very cool. Awesome. So 40, 30, 20, 0, 0. So here's one example of such a long but high frequency haplotype. So basically notice that, this haplotype carrying this European version of the lactase gene is in fact, very long but low frequency in the African haplotype. But in Europe, it is very long, but also super high frequency. So 77% of European chromosomes contain this new version, this young version of this lactase gene. So this gene encodes lactase, and it's required to digest milk. So in Europe, with the advent of agriculture, there was a mutation in the non-coding regions of this lactase gene that causes that gene to remain active into adulthood. So if you are lactose intolerant, that basically means that you are not a mutant, that you have one of those haplotypes. If you are lactose tolerant, that means that you acquired a mutation while your ancestors lived in Europe that was selected because cow's milk and goat's milk and other milk was in fact a great source of energy. Whereas traditionally, before the advent of agriculture, having the adults be able to drink the milk of the mother at the detriment of the child was a very bad idea. So it's actually advantageous to have adults not being able to metabolize milk in their adulthood, except if you invent agriculture and domestication of animals, and that basically leads to this long haplotype. So the evidence of positive selection in the European lactase gene is the persistent haplotype is present in 77% of Europeans at high frequency, and yet extends to 1 megabase, which is enormously long, as anyone carrying it was selected and it didn't have time to accumulate recombination events. Okay, so let's see who's with me so far. Awesome, very cool. So 70, 20, 10, 0, 0, 0. Okay, so we basically can now combine these multiple signals of natural selection. We can basically have these long range correlations, these high frequency derived alleles, this high differentiation between population, high frequency derived allele from chimp, and then this long range correlation in these long haplotypes, and basically combine these scores together to infer regions that are under selection. So when you do this with multiple tests together, you basically have a composite test that sort of allows you to narrow down the region of selection very carefully, very closely. And that allows you to now start identifying specific genes that are under selection in recent human history. So people are like, oh yeah, humans are not evolving. Well, there's many locations where we see clear signs of selection for a new allele that is at very high frequency, but also in a long haplotype suggesting it recently rose in frequency. And if you ask, what do these genes do? So again, lactose tolerance with the domestication of animals, resistance to malaria, by having this sickle version of your cells in a heterozygous setting, because the homozygous leads to malaria, to sickle cell disease, but the heterozygous leads to protection from malaria. And then again, there's many additional locations that are indicative, for example, of selection for height in Northern Europe for longer, for taller individuals in the steppe ancestry populations, but in fact selection for shorter individuals in the farmer populations. So this could be perhaps environmental adaptation, or this could simply be mate selection. Maybe tall people prefer to have tall partners, and that's why you basically have the selection for taller, and short people prefer to have short partners. So if there's an initial drift, it might actually be driven by sexual selection. So the rest we've actually covered during the population genetics lecture, so we don't need to cover those. So what we talked about today is how to build these genome assemblies, how to build whole genome alignment, and then how to look in closely related species, in very distantly related species, and within a particular population. So last question, who feels that they've learned something today? So very, very cool. Awesome. So 70, 30, 0, 0, 0. And then there's a question in the chat. Oh, thank you. Thank you. Awesome. Thank you, guys. See you. And go vote. Go vote as if your life depended on it, because it actually does depend on it. So please go vote. You guys are adults. You should go vote. Bye.
Sorry, Professor.
Oh, yeah, of course, Kathleen.
I'm wondering if you have received my email on the